Alright guys, here we are with chapter 27. We're talking about bacteria and archaea. So we are discussing 2 out of the 3 major domains of life here on planet Earth. We're going to find out that things have changed quite a bit over the last, say, 20 or 30 years. Molecular studies and genetics have allowed us to kind of put these into the proper categories or into more proper categories. We used to think of these microscopic creatures all in one big group called Monera. It was a kingdom Monera, but that kingdom no longer exists, and some of the new technology that we gained in the last 20 or 30 years has allowed us to study these quite a bit more and realize that what we thought was one big group of creatures is actually quite different from each other. So now they have been subdivided out, and as a matter of fact they have been given elevated to domains. So we have the 3 domains of life include bacteria, archaea, and eukarya, and we're going to look at these first 2, bacteria and archaea. Both of these, bacteria and archaea, these domains are made up of prokaryotes. So these are all prokaryotic creatures. None of them have the types of cells that a human or a dog has. None of these cells have a true nucleus or the organelles or anything like that. Most of these creatures we're going to find out are unicellular, but there are a few multicellular ones that kind of work in colonies or groups, but we'll talk about those more in just one second. Your book describes these 2 domains of creatures, bacteria and archaea, as masters of adaptation, and that's certainly true, especially with the archaea. They can be found in amazingly difficult environments that you would never find more complicated forms of life like eukarya. So, for example, in Utah's Great Salt Lake, which can reach a salt concentration of about 32%, which is 10 times that of seawater, you can find some of these living creatures. And if you've ever accidentally taken a gulp of the water when you go swimming down in Galveston or Corpus Christi or someplace like that, you know how salty the ocean water is. Imagine 10 times saltier than that. Virtually nothing can exist in that water, nothing can survive, but there are various forms of archaea and bacteria that live in those waters. As a matter of fact, there is a pink color that sometimes shows up in the Great Salt Lake, and that pink color doesn't come from minerals or inorganic materials. That pink color comes from the living creatures, the archaea and the bacteria that thrive in this high saline environment where virtually nothing else can live. So to describe prokaryotes as adaptable would be totally an understatement. They thrive almost everywhere, including places that are too acidic or salty, cold or hot for any other organism to survive. Most prokaryotes are microscopic, but what they lack in size they make up for in numbers. The vast majority of life on our planet are of the bacteria and archaea, the prokaryotic creatures. There are more of them in a handful of fertile, nice, good garden soil than in the entire number of people who have ever lived on planet Earth. So in one nice big scoop of soil you can find more prokaryotes than you have ever had human life on our planet. And I don't mean just living now, I mean all of the number of people who have ever lived. So that is an amazing fact. Prokaryotes are divided into the 2 domains we already discussed, bacteria and archaea. And what's most amazing about these groups of creatures is their ability to adapt. And that ability has allowed them to create amazing diversity and to inhabit amazing environments where you wouldn't find other types of living creatures. I didn't enclose the video inside the PowerPoints here, but if you want to go see some of these videos, they are in the notes folder for chapter 27. A good example of one of the environments where this archaea and bacteria may live is around ocean floor thermal vents, where you have volcanic openings in the ocean floor and you have superheated water and chemicals that are rushing up from the Earth's crust. The water and the temperatures can be over the boiling point of water, and so you have water basically in steam boiling out of the ocean floor. This is a place where virtually no other creature could live except for the archaea and perhaps some of the bacteria. And so there are some other creatures that live near these vents but not right inside the vent. And so this video was of some tube worms that lives near some of these other vents, and you can take a look at that. Most of these videos are only 3 or 4 seconds long, just very short clips, but again you can find them inside the folder for chapter 27. We're going to start right here with concept 27.1, and it talks about the structural and functional adaptations contribute to prokaryotic success. You're going to find that there is an amazing number of varieties in these two large domains. Earth's first organisms were likely prokaryotes, and although most prokaryotes are unicellular, there are some species that form colonies of attached cells that tend to work together to get their various goals accomplished. Most prokaryotic cells are quite a bit smaller than eukaryotic cells. Most of the prokaryotic cells are 1/2 to 5 micrometers, where the eukaryotic cells tend to be 10 to 100 micrometers in size. So you can see that the prokaryotic cells are usually quite a bit smaller, and prokaryotic cells have a variety of shapes. There are three main shapes that we usually see. The most common three main shapes are spheres, or cocci, rods, which are bacilli, and then spirals, which we'll see in just a moment. So here are the three main shapes usually that you'll see: the cocci shape, which is the spheres, the round balls; the rod shape, the bacilli shape; and then here are the spiral shapes. This is usually the form that you'll see with the archaea or the bacteria. I think most people are probably familiar with the rod shape, especially in the bacteria, but you can find all three fairly common. Another important feature that kind of defines what it means to be a prokaryote is most or nearly all prokaryotic cells have a cell wall. It maintains the cell's shape, protects the cell, it prevents it from bursting in hypotonic environments. And you may recall that there are other creatures that have cell walls too. There are eukaryotic creatures that have cell walls, such as a plant or a fungi, but the makeup of the cell wall in a prokaryotic creature is a little bit different. In the eukaryote, the cell wall of a plant is made up of cellulose, and in a fungi it's made up of chitin. Neither of these materials are used in the cell wall of the prokaryotic creature. The bacterial cell walls contain this long word here, peptidoglycan. Man, that's a tough one to say. Peptidoglycan. There we go. Say it 5 times. Peptidoglycan. Peptidoglycan is a network, kind of a cross-linkage of sugar polymers and polypeptides, and that's what's used to form the cell walls of most bacterial cells, prokaryotic cells. Most prokaryotes do the same thing as a plant cell would in a hypertonic environment. They lose water and plasmolyze. If you remember, we talked earlier about plant cells, how if you put them in a salt solution, they lose water through osmosis and they shrivel up and the cell membrane pulls away from the cell wall. Well, the same thing happens to prokaryotes too that have a cell wall, and that's why salts used to be, or sometimes still are, used as good food preservatives. People used to salt their meats, or say for example, think about pickles that are very salty. They're well preserved because that salt solution usually causes the prokaryotic cells to plasmolyze, and they can't live, or they can't effectively reproduce very well. And so that's why hundreds of years ago, before we had refrigeration, we would salt the meats, and that would act as a kind of a barrier against some of these prokaryotic bacteria and archaea. So the cell walls of a bacteria contain peptidoglycan. The cell walls of archaea are slightly different. They contain polysaccharides and proteins, but they don't have the peptidoglycan, so they're slightly different. When we are looking at some of these creatures, especially the bacteria, we have a method called gram staining that helps to kind of classify and differentiate them. Gram staining is named after a scientist whose last name was Graham. That's usually what we do in science. You do something useful after you die, we name something after you. So this gram stain is used to classify bacteria based on the cell wall composition. We're talking about bacteria for the most part here in this case. In gram staining, what you do is you take your bacterial sample, you stain it with a crystal violet stain, a bright purple type of stain, and iodine, and then you rinse it with alcohol, and then you stain it again with a red dye such as safranin, and then that's going to give you a certain color based on the composition of the cell walls. If the bacteria you're looking at are gram-positive, the gram-positive bacteria have simpler cell walls with a large amount of peptidoglycan, and you'll see that that's going to turn out to look purplish in color. It's going to hang on to that crystal violet stain. If the bacteria is gram-negative, they have less peptidoglycan in their cell walls, and the outer membrane is a little bit different. As a matter of fact, the outer membrane of the gram-negative bacteria contains some materials that are sometimes toxic to the creatures that the bacteria are in. So the gram-negative bacteria, if you're infected with them, they tend to cause symptoms such as fever and shock because of the toxins that their cell walls release. So here's an illustration of a gram-positive bacteria. It's got a more simple cell wall, heavy with peptidoglycans. The peptidoglycans trap that crystal violet stain, and so they will show up as purple when you look at them in the microscope. And the gram-negative bacteria, they have a slightly different format for their cell walls. They don't have as many of the peptidoglycans, and they have an outer sort of membrane of lipids and polysaccharides that doesn't hang on to the crystal violet stain, but it does show the red stain that we use. So if you look at an actual picture after gram staining, you can see some purple dots. Those would be your gram-positive bacteria with a heavy peptidoglycan cell wall. And then the little reddish colored dots are the gram-negative bacteria. They don't hang on to the purple stain. It gets washed off and reveals the red stain that you applied next. So this is a quick way of basically classifying or dividing bacteria into two different kinds based on the type of cell wall that they have. Of course, you might want to ask, why is that important? What does that matter? Well, we do this classification because the type of cell wall can affect how we treat the bacterial infection or how we can fight off the bacteria. A lot of antibiotics target that peptidoglycan cross-linkage of the cell wall, and they damage the bacterial cell walls. So that means the cell walls break open and the bacteria die or can't effectively reproduce. The cool thing is these antibiotics, they're going after this peptidoglycan cross-linkage, and because human cells don't have that cell wall, don't have that peptidoglycan, they're not going to be damaged. So you can take an antibiotic that will damage the bacteria cells, but it won't damage your own human cells. Now the gram-negative bacteria, they are generally a little bit more antibiotic resistant because they have this lipid portion of their cell walls that makes it harder to get the medication into them, that makes it harder to get the antibiotic to affect them. And the gram-negative bacteria also tend to cause more problems related to toxicity because their cell wall releases certain chemicals that can cause fever or shock or something like that. So it's helpful to know if the bacteria you're dealing with is gram-positive or gram-negative. That affects how we treat it or how we might go up to fight that particular bacteria. In addition to cell walls, a lot of prokaryotes have a sticky polysaccharide or protein layer called a capsule that covers the entire cell, kind of as an extra layer of protection. It's also a layer, kind of a sticky layer, that allows it to either stick to a substrate, stick to another cell, or stick to cells within the colony that it's forming together with other bacteria. So like in this example, you have a bacterial cell. You can see the cell wall, this kind of thin little ring, and then you see all of this. It kind of looks like Velcro. That's the bacterial capsule, and it's allowing this bacteria cell to stick to this tonsil cell. So this would be like a human tonsil cell, and it's infected with bacteria, and the bacteria is sticking to it using the capsule, this outer, to me it kind of looks like Velcro or fuzzy sticky material on the outside. Some prokaryotes also have other sort of appendages that stick off called fimbriae or fimbriae. Fimbriae are hair-like appendages. Also, they allow the bacteria to stick to where it needs to stick, to the substrate or other individuals in the colony. So here you have a great example of fimbriae sticking off of this creature, and this would be kind of like sticky threads that they can use to stick to other bacteria or to stick to the substrate that they are growing on or working with. And then of course there's one more that I don't have a picture of, but it's called a pili, and we'll see a picture of it a little bit later. A pili, or a sex pili, this is usually similar to a fimbriae, but it's usually longer and there's not as many of them as a fimbriae. And this is basically a tube that allows prokaryotes to exchange DNA. So we'll talk about that more in just a couple of minutes, but some bacteria will form what's called a pili, and that's basically this long tube that can connect bacteria cells to each other where they can exchange DNA. Another way to categorize or study some of these bacteria and archaea cells is their motility. Motility is basically their ability to move around. Can they move around? How do they move around? In a heterogeneous environment, in other words, an environment that's not all the same, a lot of bacteria exhibit what we call taxis. Taxis is the ability to move toward or away from a stimulus. So they may sort of swim or move toward nutrition or nutrients, or they may swim toward or away from oxygen, depending on if they're aerobic or anaerobic. Chemotaxis is a movement toward or away from a chemical stimulus, and some of these little creatures can actually move pretty well, move pretty quickly. Some of them can move at a rate that's about 50 times their body length per second, which doesn't sound like much because they're so tiny, but if you scaled that up to a human, that would be the equivalent of a human running about 190 miles per hour. Again, we have some videos. If you go take a look in the folder for this chapter, you can find some videos that I've posted, just short little videos showing motility of various bacteria and archaea cells. Most of the bacteria that can move around, they do so using something called a flagella, and a flagella is like a little tail, a little whip-like projection. Some of them have only one or two, some of them have multiple of them scattered about the surface of their body. Sometimes they're concentrated just on one end of the cell. The flagella of the bacteria, the archaea, and there are even some eukaryotes that have flagella, they're not all the same. They're actually composed of different proteins, and they likely evolved independently. So even though all three of the domains of life, you can find flagella. You can find them in the bacteria, the archaea, and the eukaryotic cells. They all developed, it seems like, at different times and in different ways. They're built from different materials. So we would call this flagella analogous and not homologous. So even though all three domains have it, it doesn't seem like it came from a common previous ancestor. It seems like they all developed independently. So this would be analogous structures as opposed to homologous structures. Some of the differences between, say, like a eukaryotic and a prokaryotic. The prokaryotic flagella are only about 1/10 the width of the eukaryotic. The prokaryotic flagella are not covered by the extension of the plasma membrane like they are in the eukaryotic. And the molecular composition and the way that they are used to move about differs as well. And then if you compare the bacteria and the archaea, there's differences there too between the flagella of the bacteria and the archaea. So again, it seems like they were all developed independently of each other, and that would make them analogous and not homologous. Again, another video, if you look inside the chapter 27 notes, should find a prokaryotic flagella showing this particular little bacteria moving around. Great little illustration, and then a microscope picture. This looks like maybe a transmission electron microscope picture of the prokaryotic flagellum. It actually acts like a little motor, and so there is a variety of systems in here that allow this little piece here, the hook and the motor and the rod, to twist, and as it twists, it kind of spins this little corkscrew shaped tail that moves it back and forth and allows the creature to move through usually some sort of liquid substrate or some liquid material that it lives in. The bacterial flagella are composed, like I said, of three parts. We call them a motor, a hook, and a filament. So this would be the motor portion right here, this piece, and this would be the rod, and this is the hook, and then this is the filament. And it's a pretty complex piece of machinery for such a tiny little living creature. Now, of course, this little creature has been evolving for billions of years, so evolution certainly has had the time to create such a interesting and complex device. A lot of times you'll get arguments against evolution using the flagella as an example of irreducible complexity. You'll get the flagella, and some people will talk about the camera eye, the complex eye that, say, a human has or a dog has, or even other creatures like an octopus who develop that eye separately from us. So some people point to these very complex structures, and they talk about something called irreducible complexity, meaning they don't see how this could come into being piece by piece by piece. But if you carefully look at the flagella, the proteins that make up the flagella of this bacteria are really modified versions of proteins that perform other tasks within the bacteria. It's not a brand new thing that suddenly just popped into being. It happened in a stepwise fashion. Of the 21 proteins required by all species that have been studied to date to make flagella, 19 of those proteins are modified versions of proteins that have other jobs, that perform other tasks within that same bacteria. So flagella likely evolved as existing proteins were added to ancestral secretory systems. Much of the proteins that make up the flagella are modified versions of proteins that have a different job in the secretory system of the bacteria. So there certainly are ways in which one portion becomes changed over time. And remember, we've talked about this word exaptation. This is a great example of exaptation, where existing structures take on new functions through descent with modification. If you want more details on this, take a look on text page 5 559, text page 559, and it goes into a little bit more detail about the flagella and how it has evolved over time. Let's take a quick break right there. We'll go into, in just a moment, we'll come back and talk more about, a little bit more about the bacteria and the archaea as far as their DNA and how they're made up and that type of thing. But we'll take a break, come right back.